Uh, we are discussing about genetic engineering now. So today particularly I will focus on fundamental methods of genetic engineering. Here I will be mostly focusing on different tools involved in genetic engineering, such as restriction enzymes, DNA ligase, plasmid, and a variety of host cell. So if you see, genetic engineering started, I would say, after 1970s. Part of the reason is this is genetic engineering. So you have to somehow cut and paste our gene for the engineering purpose. And now the problem is, as you know, the dimension of DNA in between two nucleotides, we have only 3.4 angstrom gap. So as a result of that, conventional engineering tool we cannot use to cut DNA precisely at a specific sequence. It is almost impossible. So as a result of that, like only after the discovery of few basic ingredients, for example, restriction enzymes, that can only precisely cut DNA in a specific sequence. And then DNA ligase, its utility, how it works. When we came to know all those things together, then the field of genetic engineering evolved. And then I will be also discussing about the application of gene cloning and transformation. So particularly restriction enzymes. So those are also commonly called as restriction endonucleases. So they are naturally produced in bacteria. So like particularly I would like to mention here that these enzymes are present in some bacteria, not present in all bacteria, but they are helping bacteria for their defense mechanism. So if you see here, restriction enzyme is, they are part of you can say that they are kind of their immune system. So as you know, phage virus, that means bacteriophage. So they attack bacteria. So they attack bacteria and they multiply inside the bacterial cell. So now this bacteria, they have some weapon. For example, this restriction endonuclease they have, and this enzyme digests this foreign DNA. So what happened when phage virus attack bacteria? So they inject their DNA, phage viral DNA, inside the E. coli cell, for example, or any other bacteria. Now this enzyme will degrade this foreign DNA. So as a result of that, they cannot multiply inside that bacteria, or they cannot kill the bacteria. So this is some kind of their own strategy to save them. But we are utilizing this enzyme for our own good. So now if you see, as I already mentioned, that they destroy bacteriophage DNA in bacterial cell, and also it is very important to note here that they cannot digest host DNA with methylated C. That means the question is coming now, that same enzyme present in bacterial cell, and bacteria, they also have DNA, but then why their DNA is not getting digested? So in bacteria, there are different types of modification system. They modify their DNA in such a way that this endonuclease, they can understand. So they will not cleave, they will not digest their own DNA. They will only mount response against the foreign DNA, for example, in this case, phage virus DNA. Okay. And now, if I mention a little bit here, the restriction endonuclease. So they are somehow restricted by DNA sequence. This enzyme will not cut everywhere. So this enzyme will specifically recognize a certain sequence. And there are different types of restriction enzymes are available in bacteria. And nowadays, those enzymes are available commercially also for our own, molecular cloning purpose in laboratory. We just, purchase those from some company. That's all. But the thing is, if you see from the name, that endonuclease. So nuclease means something which will cleave nucleic acid, that is nuclease. Endonuclease means, say for example, if you see this is DNA double strand, right? So I'm just putting the direction here. So if this is DNA, then exonuclease, they will cut from the one side. They will cut DNA, chew DNA from one side. But endonuclease cut from the inside. Okay, that's why their name is endonuclease, restriction endonuclease. Now, what is their substrate? Already I mentioned their substrate is DNA. And they recognize specific nucleotide sequence in DNA and cleaves the bond between two nucleotides. So bond between two nucleotides means not the hydrogen bond. Hydrogen bond is just for the base pairing in DNA. So bond between two nucleotides means the covalent bond, the phosphodiester bond present in between the two nucleotides. So that can be broken by this enzyme, restriction endonuclease. So now, based on their action, there are many types of restriction enzyme. So if we classify them broadly, like we would say blunt end cutter and sticky end cutter. So for example, if I say this is one DNA here, right? So now if you see here sequence C, 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 G, G, G. So complementary sequence should be G, 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 C, 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 and then here it's going like this, 3 prime. I am not writing the whole sequence here, 5 prime, and here 5 prime, 3 prime. So this is a recognition sequence of a particular enzyme, and this enzyme is known as SMA, SMA1. So this enzyme name you don't need to remember. This name, like are coming from where they were isolated, like from some bacteria, and their initial letter, all those things. But it is not important to remember this name. But what is important here, that this enzyme, they cleave this DNA. So this nucleotide, they have phosphodiester bond in between them, right? So now it will cleave here. So as a result of that, this enzyme will work on this sequence and it will cleave this DNA. That means it will break two phosphodiester bond, one here and the second one is here. So two phosphodiester bond will be broken. So as a result of that, what will be the end product? You will get something like this, 5 prime, C, 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 3 prime, 3 prime, G, 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 5 prime. So these two fragments are produced now. So that's why this is called blunt end cutter, this enzyme, SMA1, blunt end cutter. Because as you can see, their end is blunt, not the sticky end. When I will be explaining sticky end, you will be, you will understand even better. So let me give you one example about the sticky end. So now here, if I say this is another sequence I am writing here, 5 prime, G, A, T, T, C, and then 3 prime. Similarly, 3 prime, you have some sequence here. I am not writing, just C, T, T, A, G, 5 prime. So this is another, portion of DNA. I just, wrote the sequence here. Now, if you see another enzyme, EcoR1. This is another very common restriction enzyme we use in our lab, in every lab I would say. So this enzyme will cleave in this way. So here it will break one phosphodiester bond and here it will break another phosphodiester bond. So as a result of that, what will be the product? Now if you see, the product will be something like this. So 5 prime G and then 3 prime C, T, T, A plus here A, T, 3 prime, and then you have G 5 prime. So here you can see that their end here, this part is, that's why we say sticky end, because again this end can be joined together because they have some complementary sequence overhanging, right? So as a result of that, you can see this A, T, T. So they can again form base pairing with this T, T, A. So they are complementary to each other. So that's why they are called sticky end. Particularly this enzyme like sticky end cutter, we use very often in our laboratory for this purpose, for genetic engineering purpose. Blunt end cutter also can be used for some special purpose, but sticky end cutter is very common. And as I already mentioned that these enzymes are available as prepackaged kit and you can buy it commercially. Now I should mention that the discovery of restriction enzyme and understanding of restriction enzyme is very important for genetic engineering. And as you can see that these scientists, Werner Arber, Nathans and Hamilton Smith, they received Nobel Prize in 1978 in physiology and medicine for their work on restriction enzyme. Without restriction enzyme, we cannot do genetic engineering. So this is very important. Now, the next tool. So restriction enzyme we just discussed. The next one is ligase. So I mentioned, so in genetic engineering, we have to cut and paste DNA so that you can, modify some DNA. You can add some, you can add a gene to somewhere in a different cassette of DNA. So as a result of that, not just you have to cut it, then you have to paste it also. Restriction enzyme you can use to specifically cut a DNA segment. But now you need some other tool to paste them also. So that is here ligase. So DNA ligase, it is an enzyme that can link together DNA strands that have double-stranded break. That means a break in both complementary strands of DNA. For example, when I just explained in the previous slide that how restriction enzyme is breaking the phosphodiester bond in DNA, you notice that restriction enzyme, they are breaking two phosphodiester bond in both the strand, right? So as a result of that, one phosphodiester bond in each strand. So total two phosphodiester bond they are breaking. So as a result of that, this double-stranded break can be ligated again by this enzyme, ligase. And that's why this enzyme is called ligase. And naturally, this enzyme, it has role in DNA replication. As you can remember, in the first week, we discussed about replication. That time I told that how ligase finally fill the gap between short segment of DNA. This is the function of ligase naturally. And for this work, it needs ATP, adenosine triphosphate, for energy. And here also, like DNA ligase has extensive use in molecular biology laboratory, as I already mentioned, for genetic engineering experiment. So as a result of that, for example, if I now mention another, I'm giving you another sequence here, like 5 prime. So some sequences are present here. Then I would say G, G, A, T, C, C, and some more sequence here, and that's all. C, C, T, A, G, G. So again, this is our DNA, and we are showing only a portion of sequence which can be recognized by one enzyme that is called BAM H1. This is also very common enzyme. Very often we use this enzyme. And this enzyme will cut here. So as a result of that, after digestion, if you treat this DNA with this enzyme BAM H1, then you will get something like this. G, C, C, T, A, G plus G, A, T, C, C, G, 5 prime. So this is done. But now, as I mentioned, they are sticky end. So as a result of that, so if you mix this fragment in higher concentration, what will happen? So they can again come to each other, right? They can again fall back, like based on their sequence. So as a result of that, you will be seeing something like this: 5 prime, G, 3 prime, C, C, T, A, G. Then you have here G, A, T, C, C, 3 prime, G, 5 prime, something like this. But here the problem here is, see here this bond, they are like, this is not, the phosphoroester bond is not yet formed, because that has already been clipped by ligase, clipped by restriction endonuclease. So as a result of that, here you have to add ligase enzyme. So as a result of that, then it will form this phosphoroester bond, and it will happen in presence of ATP, or adenosine triphosphate. Now, this is very important slide, that restriction enzymes and ligase are the key players in genetic engineering. So this cut paste mechanism you can do if you use properly these two enzymes, restriction enzyme as well as ligase. I am giving you just, I am trying to explain you by some example. Let's see. So if this is DNA, and here I am putting this sequence here: G, A, T, T, C. Similarly, here C, T, T, A, G. So 3 prime, 5 prime, 5 prime, 3 prime. So now this DNA, if I treat that with one enzyme called EcoR1, I already mentioned. So if I digest this DNA with EcoR1, and I am assuming that this EcoR1 site is not present in the rest of the DNA, only it presents in one portion here, whatever I mentioned. So that you have to check during the cloning and genetic engineering experiment. So as a result of that, what will happen? So after digestion, you will get 5 prime, this fragment, then it cut here. So as a result of that, G, and then C, then T, T, A. So this is the big fragment here. So I am just mentioning here, this is the large fragment, and this side is small fragment. Okay. Now I am not writing the sequence again and again to save some time. So now this is the small fragment. Now you can actually separate these two fragments based on some other technique that you have already learned. For example, gel electrophoresis. So if you, after this digestion, if you run this product, like the digested product, in agarose gel electrophoresis, then what will happen? So this is the agarose gel. So this is negative side, this is positive. So you loaded your sample somewhere here. So after the gel run, you will be seeing that one band is here, one small band is here. So this is the large fragment. Now the thing is, you can load here some ladder that the molecular weight you know. So on the basis of that, you can actually detect your fragment of interest. Now the question is, if this large fragment, if I say this is the fragment A, now you have another DNA molecule. I am not writing the sequence here. So this DNA, you have also cut site, the same cut site for example, eco R1 cut site. So as a result of that, you can digest this DNA with this eco R1 enzyme. Then again, you will get 2 fragment, right? And now just run a gel, and again you purify these fragments, and as a result of that, finally what you can do, you can take this fragment, like after purification, for example, fragment B, and here you have fragment A. So you just mix this fragment A and fragment B in presence of enzyme ligase. So as a result of that, you will be seeing that A, this is A part and this is B part. Just for representation, I am drawing this way. So this way, now A and B should be attached together. So this A and B, they are coming from different DNA, right? But you just use the same enzyme so that they have same sequence as their sticky end, and then again those sticky end can attach to each other based on their best complementarity. And now if you add ligase, as I mentioned here, then A and B should be attached together. This is the basic technique of genetic engineering. Then only little bit of more complexity, more modification, that's all. But this is the basic idea, how you can cut and paste DNA molecule together. So you have to select properly restriction site, and then you have to design your experiment that way, because you have to be careful that the same sequence is not present in the rest of the DNA. Otherwise, your enzyme will clip here and there, and you will get many fragments, and that will complicate your experiment. So you have to be careful about that. Now, this is all about restriction enzyme and DNA ligase. Now, another very important thing is vector. So here particularly I am mentioning plasmid. So plasmid is an example of vector. So what are the plasmid? So plasmids, they are the small pieces of circular DNA used for cloning. So as I already mentioned before, like vectors, so you have to put your gene of interest inside some vector. Why? Because that vector you have to transfer in some host cell so that you can make your product, the protein of interest, for example, you can make it. I will explain all those things soon. The thing is, plasmid, so if this is E. coli, so this is bacteria, E. coli. And now E. coli, so they have their one circular DNA, that is chromosome. This is bacterial chromosome. Similarly, all other bacteria, they have this kind of chromosome or DNA. But this plasmid, they are small pieces of circular DNA, but which is extra chromosomal. That means apart from the chromosome, beside the chromosome, they have some additional small circular DNA, and this is called plasmid. So all bacteria, they don't have plasmid, but some bacteria naturally they have plasmid, and this plasmid actually help bacteria to attain some additional properties. For example, resistance against some antibiotic, sometimes resistance against some heavy metal. So there are many other function of plasmid. And as I already mentioned, that naturally some bacteria, they have plasmid, and we are using plasmid for our own interest in this genetic engineering approach. So why we are using plasmid? Why it is beneficial? I am going to explain with few points here. The first thing is self-replication. That means it can able to replicate in the host, that is independent of origin of replication. So it seems little bit complicated. The thing is, like a bacteria cell, like as I already mentioned, they have one chromosome, for example, E. coli. Now, whenever that chromosome will replicate into 2, the bacteria will divide into 2 also. So as a result of that, all the time, every bacteria, they have one chromosome only. Now, this plasmid, their replication is independent of chromosome replication. So, as a result of that, the number of plasmid can be more in one bacteria. So, as a result of that, in one bacteria might be it contains only one chromosome, but it can contain multiple plasmid inside the bacterial cell. Now, cloning site, the region containing multiple restriction site. So, a suitable vector, so I am just mentioning, say for example, this is a vector, this is a plasmid, right? So, first thing we mention that it should have its own origin of replication. This is called ORI site, origin of replication, so that it can replicate, and that is independent of chromosome replication. I already mentioned that. And now I am saying that cloning site. So, I would say if this is the cloning site, so cloning site means here you have like multiple restriction site sequence. Why? What is the purpose? So, if you have, so this is cloning site. Sometimes we say MCS, that means multiple cloning site. That means here you have some sequence which can be recognized by different type of restriction enzyme. And what is the purpose? So, as a result of that, we will use a specific restriction endonuclease and we cut this plasmid, and then we will put our gene of interest inside this plasmid. So, that is our goal. I will explain in more detail soon. Okay, so that is our goal. So, multiple cloning site. So, natural plasmid, they their sequence, you might not get many restriction site there. But the thing is, nowadays whatever plasmid we use in lab, those are kind of engineered plasmid. So, this region like cloning site is engineered in such a way you have option of many different types of restriction enzyme you can use, and you can cut that particular sequence. So, this is little bit engineered version of plasmid nowadays we use. Now, the promoter. So, promoter and operator, all those things are important for gene expression, as you already know, right, to support the expression of insert DNA. For example, you are interested to make some protein, and you are actually the gene of interest, whatever you are working on, that gene of interest you can put in this plasmid cassette, and then if you are putting it properly under some promoter, so that in right condition this gene will be expressed into RNA, mRNA, right, messenger RNA, and this messenger RNA will be converted into protein also. So, translated into protein. So, as a result of that, you should have promoter, operator, all those sites for proper function. And then selectable marker. So, this is also very important. So, selectable marker, for example, like some antibiotic resistance gene, for example, ampicillin resistance gene. So, ampicillin is an antibiotic, and this should be present in the plasmid itself, right? And then this ampicillin resistant gene will provide resistance to that bacteria. And then proper size for easy handling. So, if it is too big, then it is little bit difficult for handling. So, that is why another important thing is like the size should be somewhere it will be more useful, right, not very big or not very small. So, this is your promoter site which I mentioned before. So, these are the basic features of plasmid which are very beneficial for our genetic engineering approach. And then host cell. So, our goal is, so for example, you are interested on some protein. So, corresponding gene you clone inside the plasmid, as I already just mentioned about the plasmid. You just put your gene of interest in plasmid. Now, the question is like you have to express the protein. So, as a result of that, you have to transfer this plasmid into some host cell. Very frequently we use like laboratory strain of E. coli, Escherichia coli. This is the laboratory strain, non-pathogenic strain. So, you can use this E. coli strain as host cell to express your protein of interest. So as a result of that, whatever engineered plasmid, that means plasmid with your gene of interest, you have to transfer that into the E. coli cell. And then if you grow E. coli cell and if you put proper condition, E. coli cell will make protein for you, because that protein coding genes is already present in plasmid. That is the overall idea of this, experiment. Apart from E. coli cell, we can use different type of cell. For example, this is prokaryotic expression system where we use E. coli, and many protein we work with, like eukaryotic protein, membrane protein, they might not express in E. coli cell, or they might not hold properly, right? So as a result of that, many times we need to use eukaryotic expression system. Many times we use yeast expression system, like that yeast is eukaryotic cell like fungus, and then we use sometime insect cell line, right? For example, Drosophila S2 cell line. This is very commonly used for eukaryotic protein expression system. And on top of that, like we have different types of mammalian cell line as well. So all it depends on your application, what kind of protein you are interested on, and whether you can express in bacteria or not, then you have to rely on some other system, right? So bacterial expression is much cheaper. That's why sometimes we start with bacterial system. But the thing is, if your protein is, it require like, lot of post-translational modification, which is not present in bacterial system, then you have to rely on eukaryotic expression system. Okay, so this is all about different types of host cell can be used for different application. Now, cloning and transformation of a gene. So here, like we are trying to like explain you whatever I mentioned in a brief way. So as you can see, this is the plasmid, and it contains ampicillin resistance gene, and this is the DNA fragment, as you can see. Now you can cleave this plasmid, right, with a particular restriction enzyme, and the similar set of enzymes you have to use to process this, like to get this DNA fragment as well. So as a result of that, finally you will be getting this recombinant plasmid. Why it is called recombinant plasmid? Because this plasmid is not the natural plasmid anymore. You put your gene of interest here, the recombinant plasmid. Now this plasmid you have to use for transformation. Transformation means like E. coli cell. So you have this plasmid, and you have your gene of interest here, gene of interest. And now if you transfer this in E. coli cell, so this E. coli cell, so it has its own chromosome, but this E. coli cell is not antibiotic resistant. So as a result of that, this E. coli cell, if you put in some media, if you grow this E. coli cell in some liquid media, for example, then if that media contain antibiotic ampicillin, for example, then this bacteria will not survive. But now if you have this plasmid inside this bacteria, so this plasmid, it has ampicillin resistance gene. So it has ampicillin resistance gene. So as a result of that, if the plasmid is present inside this bacteria, then this bacteria will survive in presence of ampicillin. So that's why you can actually select that in which E. coli cell you have your plasmid, and intentionally you put your gene of interest in the plasmid itself. So as a result of that, whenever the plasmid is going inside the cell, you are also transferring your gene of interest. So that is what you can see here, that after this transformation, like you can see, this is transformed cell, transformed E. coli cell. It has the plasmid. As you can see here, this is plasmid. So as a result of that, it will survive in presence of ampicillin. But this one, so this cell, so you cannot see any plasmid present here. So as a result of that, this cell will die in presence of antibiotics. So as a result of that, here we are using antibiotic as antibiotic for selection purpose. So we want bacteria which contain plasmid, and already you engineered the plasmid in such a way that every plasmid containing your gene of interest. So as you can see here in this cell, this is the DNA, the bacterial DNA, and you have plasmid in this bacteria. So now if you grow this bacteria, so bacterial division cycle is very fast, right? Very quickly bacteria will multiply. And if you keep your antibiotic concentration to some in such an extent that the bacteria will survive only if they have lot of antibiotic resistant gene. And since antibiotic resistance gene are present in their plasmid, so they will replicate their plasmid, they will try to keep the number high, the plasmid number high, and so as a result of that, they can defend this antibiotic against this antibiotic ampicillin. And if the number of plasmid high, that means the gene of interest is also present in every plasmid. So that is the overall idea. So in every cell you have multiple plasmid, and all these plasmid, they contain your gene of interest. And now you can express your protein of interest. So you have to for example, when we discuss about lac-operon, I mentioned that some inducer can be added. So similarly, you can clone in such a way that you can control the expression of your protein of interest. And now this is I would say kind of little bit of application purpose, the production of a protein of interest through genetic engineering approach. Whatever I mentioned on the based on that discussion, I am going to say that many proteins, even many human proteins, they are produced in bacterial system. For example, I would say insulin. All of you know that insulin is produced in bacterial system, like human insulin I am saying. And then many growth hormone, and another example I would say erythropoietin, So all these proteins can be produced using recombinant DNA technology or genetic engineering. Then the advantage here is, see, for example, insulin or any other human protein in one cell in our body, in one cell, this information to make this product, how many times is written? Only twice. So we are actually deployed. So we have 23 pairs of chromosome. So as a result of that, in mostly I would say, if I say gene A, just I am giving one example, gene A, it is present in both the pair of chromosome. So as a result of that, I would say just 2 times it's present in every cell. Now the thing is, when you are cloning this gene A inside this plasmid, so now you clone this gene A in this plasmid, and now this plasmid, there are different types of plasmid. This plasmid can be high copy number plasmid, so that plasmid can exist in very high copy number in one bacterial cell. So as a result of that, when you are putting this plasmid in some E. coli cell, so this is for example some E. coli cell, so this is the plasmid here, and this plasmid contains your gene of interest here, gene A. Now when this E. coli, they are growing in some liquid media, so if you keep proper antibiotic concentration, because your plasmid is providing the resistance against antibiotic, so as a result of that, the number of plasmid will be very high. Sometimes the number of plasmid can be more than 1000 per E. coli cell. So now you can understand that even a small cell like E. coli, but it contains like 1,000 copy of your gene of interest, that gene A. So as a result of that, you will get huge amount of product. Product means here proteins. So this is the beauty of this system, that the recombinant DNA technology, that you are cloning a gene of interest in a plasmid, and then you are sending this plasmid to E. coli cell, inside E. coli cell, laboratory strain of bacteria, for example. And now you are giving instruction to make the protein, right? So that can be done. And if you properly keep the antibiotic concentration, and if you select correct plasmid, in every cell, the number of plasmid can be as high as 1,000 or more. So as a result of that, you can understand that huge amount of protein you can make, which is not possible from the eukaryotic cell. Like without cloning, if you try to purify this protein some tissue sample or somewhere, it is not possible. So this is the power of genetic engineering or recombinant DNA technology. Okay, and you can go through any of this textbook, right, for the additional understanding, and that's all.